Hello, everyone, and welcome to American Pharmaceutical Review's QC Corner. I am Mike Auerbach. I'm the editor-in-chief of American Pharmaceutical Review, and today we're going to be talking about environmental monitoring, specifically the benefits of a single incubation temperature. And with me today are Thierry Bonnevay. He's the Global Microbiological Analytical Expert at Sanofi Pasteur, and Laurent Leblanc, R&D Manager at bioMérieux. Gentlemen, thank you for joining me today. Just to give a little background on this conversation, the environmental monitoring program is an important quality control for pharmaceutical manufacturing, and the most common usage is doing sequential incubation time and temperature to maximize the range of recovery of the flora. Current practice is to start with a lower temperature to enhance the growth of environmental strains such as molds, and to switch to a higher temperature to recover human skin microorganisms. So we're going to be talking a little bit about all these factors and details today. So our first question is, what are the main challenges for EM practicing at these two temperatures?
So thanks a lot for the question. I will start to answer. So that's right, that currently in the pharmaceutical industry, depending on the area, between 40 to 60% of the pharmaceutical industry use two temperatures starting by low to high. That is to say, as you say, starting 22 during 3 to 5 days, and then switch to 32, 2 to 3 days. So the main challenge doing that, starting with a low temperature, is that starting to the low temperature, because in environmental monitoring, we still look mainly from microorganisms coming from humans, that is to say cocci gram-positive, and starting to 22 degrees could affect really the viability of this bacteria cocci gram-positive. And that it's clearly mentioned, for example, really clearly mentioned, for example, in the USP 1160, that it's a guideline, and it's really written in that chapter that the main challenge, starting by 22, is that you could compromise the recovery of the cocci gram-positive that are the main, I will say about 70 to 80% will be a cocci gram-positive recovering from the environmental monitoring. And so you can compromise this recovery because we are facing to sublethal stress bacteria that could be difficult because they really prefer a temperature more than 30 degrees.
Mm-hmm. Excellent.
And if I may, I can add just one comment on this topic. In addition of the temperature, we know that the culture media play a key role also in the capability to recover microorganisms from the environment. Today, we are speaking about especially bacteria that are the main contaminant of clean rooms. We know that the current practice today is to use casein, soy, agar usually to recover microorganisms, but we know that also other type of media such as saburo or malt agar can be used also to be more focused on molds and fungi. Today, what of the main challenge is to have the best practice in terms of environmental monitoring control, to have the best compromise to recover everything. And today, in addition of the temperature, the choice of the culture media is key also to be able to provide accurate results in terms of environmental monitoring.
Excellent. Thank you, gentlemen. Now, I understand there's been some industry efforts to improve EM programs, like the recent PDA task force initiative. Can you comment on any outcomes of this study and what PDA is doing?
So thanks again for the question. You are right. There was a PDA task force was launched about 5 years ago in 2018. And this PDA task force was also with 8 to 9 different pharmaceutical industry. And the main topic is so one media that's really now in the pharmaceutical industry, mainly it's only the TEC media used for both fungi and bacteria, and also the temperature. And this task force really tried to analyze if a one temperature could be used and not two different temperatures, so only one temperature in how many time. So starting this PDA task force, first by an in vitro study. An in vitro study was held in 2019 and 2020. And in 2020, and try to compare. So it's an in vitro, it was an in vitro study, that is to say a selection of 13 different ATCC strains, so not the exact local isolate from the pharmaceutical industry. This 13 ATCC strain was 8 bacteria, 2 yeasts, and 3 different molds, and they studied the 3 different main using conditions, that is to say, the 22 4 days following by 32 3 days. Also the contrary, that it's called low to high. The contrary, so 32 degrees 3 days following by 22 degrees 4 days, and really compare to what could be the future standard in the pharmaceutical industry, that is to say 27.5 degrees, so between 25 to 30 degrees during 7 days. And so each different company, the 8 different pharmaceutical company with the same ITCC strain that I mentioned, the 30 different strain, study these different conditions between, of course, 10 to 100 CFU on different agar plates. And they conclude during the study that the one temperature scheme, that is to say, 25 to 30 degrees during 7 days, at the end, with these 13 different ATCC strains, perform as well as 2 temperature, low to high or high to low. And that conclusion of that study was communicated during the PDA annual pharmaceutical microbiology conferences in Bethesda in October 2020. Then following to that is really interesting, an in situ study, so no more ATCC strain, but real different sample collected in C or D area to be sure to have some events. Some different microorganism on the plate was planned in 21-22, and also compare these 3 different scheme of temperature, one temperature, one single temperature during 7 days against low to high and high to low. But it was a little bit more difficult to put in place in the 8 different pharmaceutical industry this in situ study, that it's unfortunately not yet completely finalized. We are ongoing on that still. First, in the planning, it was dedicated to be handed in 22, and we hope to completely finalize it and wait for the final result. Sorry. Of course, we hope soon to have a publication in the Journal of PDA to summarize all the in vitro study and also this in situ study to, at the end, try to advocate the other pharmaceutical industry as well as the health authority that one temperature that could be 25 to 30 degrees could be in the future the main chosen condition for environmental monitoring.
Excellent. Thank you. So let's talk a little bit about what pharmaceutical companies should do if they want to explore the implementation of a single temperature approach for their environmental monitoring programs. Either of you care to comment on what steps they should take?
So I will start. Of course, it will be completed, I think, by Laurent. The main recommendation to explore is to well know first your classical and normal different strain as fungi and bacteria you have in your pharmaceutical environment. And then there is no other solution to have some data. So again, it's the data that are very valuable, because in the different regulation, there is no a real consensus about this environmental monitoring as media, temperature, and time. So it's quite open, and it's each company to decide and to design their environmental monitoring as the media, the temperature, the time of incubation, the condition of incubation. It's clearly mentioned that you have to validate this condition to be sure that it's not only used as more historical data, but real data that are validated to choose the most best condition in your own situation. And so you have to bring, as well as for the PDA task force, some first in vitro study. In vitro because the in vitro study could be more reproducible because you choose the microorganism, some slow microorganism or worst case microorganism, and then you can, of course, more master the concentration between 10 to 100 and compare different conditions. And then you have to follow by completed this in vitro study by some in situ study in the normal and classical area to see if the in vitro study is also confirmed by this in situ study. And then when you have sufficient data, then you have data to really demonstrate that you have chosen as a risk-based assessment completed by in vitro and in situ study the best condition for your environmental monitoring, to not miss any kind of bacteria first, because they are the main microorganism recovered by environmental monitoring, as well as some fungi.
Yeah, and just to complete what just has been said by Thierry, data is really key today to build the environmental monitoring strategy, to choose a good temperature, and also to be sure that thanks to the risk analysis that has been performed, there is no risk to miss anything during the different controls. Today, we did a comprehensive study to investigate the best range of temperature to implement single temperature. It was a kind of copy-paste of the PDA task force. We use globally the same protocol using a biggest panel of microorganisms, and we investigated the behavior of this different panel of microorganisms at different temperature, and more especially between 25 and 30 degrees. And we have demonstrated that this range currently is suitable for a single temperature implementation for routine. So this is something that is also a first data set of information that can also provide additional thinking to orientate the testing first of the real microorganisms, flora, to be tested in this range of temperature with a higher success of implementation in this range.
Mm-hmm. Excellent, thank you. So as you were talking a little bit about environmental monitoring, visual inspection is a manual task performed by people who have, you know, variable qualifications or you know, capabilities. So how do you and improving the quality of data from EM results seems difficult without knowing, you know, the limit of these operators. So the methodology has to be improved. So what is the limit that qualifies a macroscopic object, and at what level of size can we consider the detection accurate?
So I can answer to this question. So we asked the same question to ourselves, and we try to figure out if this answer already exists. And finally, when we asked this question, we discovered that it was not so obvious, and there was no real study that can provide accurate data regarding the limitation of the human eye when operators are looking at Petri dishes. There are some studies on broth, but on real solid media, not so many. So we decided to build our own data set, and we used, instead of microorganisms, different beads with several sizes that has been put on agar surfaces on Petri dish. And we asked to trained operators to do an enumeration, a detection of the different events. So we decided to go with various sizes from 50 micron up to 500 micron, use different contrast of beads and different position on the Petri dish. And at the end, we generated a lot of different analyses, 12 operators that read more than 1,000 plates, and it generated more than 10,000 results. And finally, we were able to demonstrate that there was a lot of variability between operators, not depending on their initial training, because globally operators are performing the same. So this is really something that is a human limitation and not a training difference. We have demonstrated that there is a kind of psychological effect also for people that are afraid to miss something inside the Petri dish, and they are more affected by the generation of false positive. So they are seeing objects inside the plate that are not present. So they are kind of imagining them. But at the opposite, they are doing a very few false negative. And you have the opposite behavior with operators that when they don't see finally object, they are losing it, and they are generating more false negative than false positive. So there is these two behaviors that are more linked to the way their brain is subjected to look at the exercise of reading inside Petri dishes. And knowing this, we were able to evaluate the different factors impact in this study, and we've demonstrated that there is a kind of threshold where the human eye is not accurate at all, and this limit is around 250 micrometer. We know that there is a dramatic decrease of the performance of the human eye when the objects on the surface of agar plates are smaller than this critical size.
Mm-hmm. Excellent. So the use of automated incubators and plate counters seems to be more efficient, right, to improve environmental monitoring processes and to give consistency in results and accuracy. So how can pharmaceutical companies implement automated solutions to replace traditional plate enumeration? Do you have any insights into that?
I will start, and then it will be completed by Laurent. So in the pharmaceutical industry, because we still be in a traditional methods based on culture on different agar, but it's more the reading and the final result. It's more an automate qualification than a real alternative method. Of course, you must demonstrate that you are still as accurate as the traditional method that Laurent just explained, that it's unfortunately really not perfect, about the different explanation about the limit of detection for some macroscopic object. And so this automation of a traditional method based on culture is more a qualification of an automate and the demonstration that you are still the same performance of your final method, and it's not a real alternative complete validation methods.
And in completion of this, today, in terms of automation, there are different technologies that exist. Some of them are based on alternative methods. Some of them are using traditional culture media that are, I would say, that are closer to what is performed in routine with operators. So depending on the technologies, there is different way to go with these technologies. Today, what we have demonstrated is that technologies that are only based on final detection on Petri dish has a level of performance that is slightly lower than technologies that are using kinetic, so that are following the growth all along the incubation. And knowing that the performance of the final result is very important to guarantee good results of the environmental monitoring routine, then we recommend today, if the pharmaceutical industry is considering implementing automated system, to look at kinetic-based instruments that should have better performance in terms of reading and enumeration compared to the other systems.
Right. Are there any other advantages for using automated colony counters that you can talk about?
Yeah, sure. So we're using automated counter can provide additional benefits that can be seen on different angles. The first, I would say, benefit, is about saving time, because I just talked about kinetic technologies. So with the kinetic reading, we have ongoing during the incubation enumeration that is occurring, and when a deviation out of specification occur, then early indication can be sent to the laboratory, and then we can act on problem earlier than with the current methodology that is globally an endpoint. At the end of the incubation, we only know the result. Here, we can have an idea of the result during the incubation. There is also some aspect regarding saving money also, because there is no need to have specific investigation when you have plates that can't be read, because there is I would say defaults on the dish, or colonies that are swarming and makes the enumeration impossible. So what we call today two numerals to count. With the kinetic system, the growth is followed from the start to the end, and then the system is able to generate a result, an enumeration and then save also time of investigation when some too numerous to count events occur. There is no manual reading of the plate. There is no transcription from the Excel file to the limbs. So this is saving money. So, in addition of this, there is also a huge benefit of data integrity. Putting in place paperless solution will help dramatically to increase the data integrity compliance. And the last but not least angle we can see on automation is about operator themselves, because we all of us know that reading petri dishes, it's not an easy task. It's not a good posture for the operators that are looking at dishes very precisely and that are doing repetitive movement. So this can also help to reduce some health problem that this task can generate. And of course, that can also limit human error because everything is performed by an equipment.
And to re-emphasize that just said by Laurent, in pharmaceutical industry, the main focus for us is a big improvement in data integrity for sure. And as Laurent explained, for the too numerous to count, because you've got some mold that could be spread all over the plate, or some swarming colonies like some bacillus that render the final reading very difficult. And so in that situation, it's very difficult to have a final accurate result. And in such situation, you've got, in fact, no result, and you should think about the worst case. With this kinetic reading by the automate, you could really go before and check everything and really have a real final result, even if you've got some spreading, some swarming, different microorganisms on the plate. And it's very key, because in class A or from some really critical samples, you have to have a final result. And of course, by manual inspection and reading on the plate, there is nobody, no technician who will read the plate every hours during 5 to 7 days. It's impossible, but it's now possible with such automate.
Right. So we've talked a little bit about several of these critical issues and topics to rapidly detect variations in a facility and establish, alert and action limits, to quickly detect problems, so to keep a facility in a state of control. So looking at all of these things, do you see an advantage of combining single temperature incubation, automated plate readers, and an ad hoc SW that can provide these trending and deviations earlier?
Yes, so we have seen that kinetic technology are able to provide early results. In combination of single temperature, we have seen in our study that higher temperature in the range of 25 and 30 degrees can also faster a little bit the time of detection of the microorganisms. Because we have seen that for the majority of the molds, incubating slightly higher in terms of temperature will not impact their detection, but will foster a little bit their growth. And it is globally the same for bacteria. So in combination with automates that are able to read in a regular manner during incubation, the possibility to reduce the time to results is something that could be achievable. So instead of having two temperature, a change of temperature in the middle of the incubation and final reading, then with an automate, it could be possible to have a single incubation temperature that is increasing the time to detection of the main microorganisms of the environment, and have also early alert. And even as the system is able to detect very, very small events, smaller than the human eye, the possibility also to decrease the final incubation time that is today mainly between 5 and 7 days, below 5 days, 4 days, or even 3 days. So this is something that can be achieved.
Excellent. So we've talked about a bunch of topics and solutions today. Our final question is, what are your recommendations, if you have any final recommendations for a company that wishes to adopt a single temperature solution and invest in a more accurate way to count colonies and have trending and earlier identifications? Any recommendations or best practices that you can speak about?
So I will start. As mentioned already, there is many advantages following implementation of such automate for the environmental monitoring reading, even if up to now there is still some challenging about the capacity of such automate, but we think that of course it will be improved in the future. And the main recommendation is, of course, we have to convince our top management, but as already explained by Laurent, there is some improvement in quality, in alert, in robust, in more accurate results in data integrity, as well as some time and so money saving at the end. And then convince your top management to invest in such technology. Then you have, of course, to bring, as I already explained, some in vitro study, selecting a very well chosen different microorganisms between 10 to 20 microorganisms, including yeast and mold, of course, including worst case and slow grower and more challenging microorganism compared to, of course, in situ study. And then you have sufficient data, of course, then make the change and the comparison with the traditional that you are at least the same performance. And of course, by the network, by the exchange, by the different pharmaceutical industry, because many pharmaceutical industry are interesting in some such automate, combined with a more single incubation condition, exchanges with the different pharmaceutical industry to combine all these raw data coming from in vitro and in situ study, and at the end, convince, of course, and advocate the different health authorities.
Excellent. Thank you. Well, those are all of our questions for our experts today. I'd like to thank Thierry Bonnevay from Sanofi Pasteur for helping us today and answering our questions, and also Laurent Leblanc from bioMérieux. Thank you very much for answering our questions. Gentlemen, thanks again, and have a great day.
Thanks.
Thank you.